Okay, Chemistry 102. This is practice exam 5. Let's get started here. I'll fill up my name and let's get to it. All right, question number 1. Which of the following statements concerning enzymes is false? Inhibitors are molecules that cause an enzyme to lose its activity. That is true. If we have an inhibitor, it's going to bind to an enzyme and do exactly what it says. It's going to cause it to lose its activity. So this is not the answer. A is true. Enzymes are the body's biological catalysts, increasing the rate of cellular reactions. That's a great definition for what enzymes are. Enzymes are usually globular proteins with flexible three-dimensional shapes. Yes, that's caused by their tertiary or quaternary structure. That one is also true. The next one, the reactant in an enzyme-catalyzed reaction is called a cofactor. That is false. It is called the substrate. So the reactant in an enzyme-catalyzed reaction is called the substrate. And so the correct answer would be D. This statement is false. Let's just check E. It says the active site of an enzyme is where the substrate binding and catalysis occur. That is absolutely true. So the answer is D. Okay, let's move on to question number 2. It deals with sucrase is an example of an enzyme that displays absolute specificity. What does the term absolute specificity indicate about an enzyme? Well, if we look at A, it says it catalyzes the reaction of only one class of compounds. That would be group specificity, so like functional groups. It catalyzes the reaction of only a single type of bond. This is called linkage specificity. It catalyzes the reaction of only a single substrate. This is, in fact, called absolute specificity. And so this would be the correct answer. The next one, D, it says it catalyzes the reaction of only a single enantiomer. That would be stereochemical. So stereochemical specificity. And the last one says it catalyzes only one type of reaction for the same class of compounds. That doesn't define any type of specificity. Number 3, which of the following describes how an enzyme can affect the transition state in an enzyme-catalyzed reaction? The first one says placing a bond on the substrate, making it easier to break. That is definitely true. If you look at sucrase, that's what it does. It places a stress on the bond between the glucose and the fructose, bringing two reactants together in close proximity and suitable orientation for the reaction. That is absolutely true. Modifying the pH by accepting or donating protons, absolutely true. And so all of the choices are in fact correct for E. A, B, and C. Each one of those describes how an enzyme can affect the transition state in an enzyme-catalyzed reaction. Next question deals with feedback inhibition. In feedback inhibition of an enzyme-catalyzed biosynthetic pathway, what frequently serves as a negative allosteric effector? And the textbook has a great example of negative allosteric effectors. And she says it's kind of like a thermostat. Like a thermostat, in that if you turn up a thermostat, what are you trying to do? You're trying to produce heat. But it is the heat that ends up causing the thermostat to shut off. And so the answer is not going to be oxygen. It's not going to be the starting material. It's not going to be temperature, and it's not going to be acetylcholine. It's in fact the final product of the pathway. Once the final product has been produced and there's enough of that product, it's going to serve as a negative allosteric effector, and it's going to shut off one of the enzymes along the pathway. Number 5. Which of the following correctly describes pepsinogen? Well, pepsinogen is a zymogen. It is a zymogen or a proenzyme, which is an inactive form of an enzyme. And so the best answer would be it is the inactive form of the enzyme pepsin, inactive form. So pepsinogen, like chymotrypsinogen, and trypsinogen, whenever you see inogen on the end as a suffix, you know that you are dealing with a zymogen or a proenzyme. Okay, well, let's move on from there. Number 6, it says proteases or proteolytic enzymes are responsible for which of the following functions? What does a proteolytic or protease do? hydrolysis of the peptide bonds between amino acids and proteins. That looks like the correct answer to me, because proteases hydrolyze proteins, and proteins are held together by peptide bonds. The next one says formation of the Zwitter ion form of a protein. It's got nothing to do with that. Hydrolysis of the ester bonds in triglycerides. No, that would be a lipase. Formation of bacterial cell walls has nothing to do with that. Formation of a glycosidic linkage has nothing to do with that. A protease or proteolytic enzyme is involved in hydrolysis of peptide bonds. Okay, next question, number 7, deals with a protease enzyme. In fact, it deals with chymotrypsinogen. It says cleaves peptide bonds on the carbonyl side of aromatic amino acids. The aromatic amino acids, in fact, that it cleaves on the carbonyl side of are phenylalanine, PHE, tryptophan, TRP is the abbreviation, and tyrosine. So those ones there. Which labeled bond in the peptide below would be cleaved by chymotrypsin? Well, you can see that we have a CH2 and a phenyl here. So this portion here, you see that it goes NCC, and then it goes NCC, and then it goes NCC again like this. The first residue, or the first amino acid, is a serine. Okay, the next one is your phenylalanine, and the one following that is aspartic acid. So aspartic acid. Anyhow, so we have our phenylalanine. Now it says it's going to cleave it on the carbonyl side of the aromatic amino acid. If it goes NCC, the carbonyl that's in my phenylalanine is right here. So here's the peptide bond that's going to be broken. I'll highlight it in green. So the answer is E. That is the bond that will be broken. That is the carbonyl side of the phenylalanine in this tripeptide. Number 8, which of the following is a medical use of enzymes? I didn't go over these in our lecture, our recorded lecture that we discussed chapter 19. But if you look in the textbook, it says here, an enzyme replacement therapy for missing or malfunctioning enzymes. That is certainly a medical use of enzymes. An enzyme assays to diagnose damage or disease to a particular organ or tissue. Yes, that's a medical use of enzymes. Analytical reagents in the clinic, laboratory. Yes, injection of enzymes to counteract blood clots. And so, in fact, all of these choices are correct. These are all medical uses of enzymes. Number 9, a really good question, because in order to get into this class, you have to take Chemistry 101 or the equivalent of that class. And it says, what is the effect of an enzyme on the equilibrium constant for the reaction it catalyzes? You guys should remember from Chemistry 101, K equilibrium is something that you would have talked about. First answer is it may increase or decrease the equilibrium constant depending upon the specific reaction. B, it makes the equilibrium constant numerically equal to the reaction rate. It increases the equilibrium constant. It decreases the equilibrium constant. And the last one is it has no effect on the equilibrium constant. Remember, an enzyme only serves to increase the reaction rate. Okay? Increases reaction rate only. It is not going to affect the equilibrium. So enzymes do not affect equilibrium. And so the correct answer would be it has no effect on the equilibrium constant. Okay, an enzyme won't affect that in any way, shape, or form. Number 10, in which class of enzymes would you assign an enzyme that carries out the dehydrogenation of its substrate? Well, if you remember, something that we talked about in the organic chemistry section of this class is loss of hydrogen, which is what dehydrogenation is. Okay, this would be oxidation, right? Because we said that oxidation is either the gain of oxygen or the loss of hydrogen. And so this enzyme, or an enzyme that carries out the dehydrogenation of a substrate, would be an oxidoreductase. An example, we'll put here an example, would be a lactate dehydrogenase, for example. Lactate dehydrogenase. Okay, that's an example of an oxidoreductase. Right, lyases, those are involved in either the addition to or the removal of groups from a double bond. Hydrolases, those are involved in hydrolysis, and transferases, those are involved in transferring groups like amino groups or phosphate groups. And so the answer would be oxidoreductases. Number 11. For enzyme catalyzed reactions, what happens when the concentration of the substrate is increased? Well, we looked at this in a graphical representation, and we said here, if we have the concentration of substrate down here, and then we have reaction rates here, what we see initially is as we increase the amount of substrate, we get an increase in the reaction rate, and then all of a sudden it will plateau. It will level off. Okay, so it says here the rate continues to increase as the substrate concentration increases. That's not true, because that would be like saying it just increases no matter what. As long as we add more substrate, the rate's going to increase, and that's not what happens. In B, the rate decreases, can't be true. In C, the rate decreases, can't be true. The last two, if you look at D, it says the rate increases until it reaches a maximum constant value, and that's what you see in this graph right here. The last one, the rate increases until it reaches a maximum and then decreases. It has nothing to do with decreasing again. 12. What portion of an enzyme is in direct contact with the substrate? Well, the substrate is only going to bind to an enzyme in one particular place, and we call that the active site. Now, enzymes are very large, right? We saw structures of several enzymes throughout the course, and again, those are really big molecules, but we saw that where enzymes bind are very particular places in the enzyme, and they're very small. Only a small portion of the enzyme is set aside for the binding of a substrate, and so we call that the active site. Number 13, an enzyme catalyzes the removal of an amino group, from one substrate and its addition to another compound. To what class of enzymes does this belong? Now, when I hear about transferring an amino group from one compound to another, I think of a transaminase. And we looked at a couple of examples of transaminase. And a transaminase would be part of a bigger group, which would be the transferases, right? For example, a kinase is also considered a transferase, but a transaminase would catalyze the removal of an amino group and its addition to another compound, and that falls under the umbrella or the class of enzymes referred to as transferases. Okay, the next question is kind of hard to read the answer. It came out. This is the question right here. It says phenylketonuria, PKU, is an inherited disease caused by the inability to produce a properly working form of the enzyme phenylalanine hydroxylase. This enzyme catalyzes the conversion of the amino acid phenylalanine to the amino acid tyrosine by adding a hydroxyl group to the para position on the ring. Based on this information, what is the structure of tyrosine? So this is the phenylalanine right here. You can see that on the phenylalanine, we have one substituent, right? It's all of this portion right here. Now it's telling us that we're going to have a hydroxyl group para on the ring. Now, hopefully you remember your positions on the aromatic ring. If we have them 1, 2 like this, we call that ortho. If they're in a 1, 3, so if we have them 1, 3 like this, we call that meta. And when they're 1, 4 from one another, I'll try to be consistent here. That's okay. If we have X and Y like this, then that is what we call para. If we're putting a hydroxyl group at this position, it's not this answer here, but it would be this answer here because we have a hydroxyl group in the para position. Now the answer is actually B. Kind of looks like it's C the way that it's written, but the answer is B. So this would be tyrosine. And you can double check me on that, but this is definitely the structure of tyrosine. I think of it as, representing the word tire swing. It kind of looks like you have a place to swing. I don't know. Okay, and then the question didn't print out quite correctly, but let's move on from there. Question number 15. of the following types of intermolecular interactions, which is not likely to be involved in the binding of an enzyme to a substrate. So if we think about a substrate binding to an enzyme, you should be able to come up with all the possible ways that a substrate would bind to an enzyme. For example, van der Waals interactions, right? Van der Waals would fall under the categories of London forces, and dipole forces. Those intermolecular forces count as van der Waals attractions, and yes, those are definitely involved in the interaction or the binding of a substrate to an enzyme. The next one is hydrogen bonds. Absolutely. Right, we talked about hydrogen bonds. Polar interactions, absolutely as well. That would almost fall under the umbrella of dipole. What else? Let's skip D. We'll go right ahead to E. Ionic interactions or ionic attractions. Yes, we saw that if we had a carboxylate, for example, and let's say we had, a protonated amine from a lysine, okay, something like this, yes, there would be an electrostatic interaction like this. But covalent linkages, no, those are not responsible for the binding of a substrate to an enzyme, because we know that substrate binding is reversible. Okay, so covalent bonds aren't going to cut it. They're going to be way too strong. They are, in fact, irreversible, or that would cause an irreversible binding. Okay, number 16. What are the two ways in which the energy of a food source can be released in a catabolic pathway? Remember, the catabolism is breaking down foods. The first answer is chemical bond energy and heat. Then we have electrons and enzymes, phosphate and enzymes, heat and light. I think this one is funny. You can imagine yourself just glowing after you eat some food, maybe. Chemical bond energy and light. Well, the ones that have light in them, that's just crazy. Okay, electrons and enzymes, phosphate and enzymes, definitely not the correct answer. The correct answer should be abundantly clear that it's chemical bond energy and heat, and that chemical bond energy is stored in ATP. So you can think of the human body in a way as just a big combustion chamber. We take in food, we burn up that food. Not only do we get energy from it, but we produce heat as well. 17. The abbreviated form of the equation for breaking the phosphoanhydride bond of adenosine triphosphate is shown below. What is the missing product in this reaction? Well, if we take ATP, which is adenosine triphosphate, and we convert it to adenosine diphosphate, well, we release some energy, right? We release 7 kilocalories per mole, but there's something else. Well, if we went from a triphosphate to a diphosphate, what we must have, or what must be missing, is an inorganic phosphate group. Okay. In fact, we went over that in the lecture for chapter 19, that you lose chapter 21, that you lose an inorganic phosphate group. Okay, number 18. The primary function of catabolic pathways is the generation of a particular substance. What is that substance? Well, the whole reason that we do catabolism is to get enough energy to allow organisms, in order to allow them to survive and to function. And so the cellular energy currency is adenosine triphosphate. And that's something that you need to know in this class. ATP is the purpose of catabolic pathways. Let's look at number 19. Digestion of carbohydrates begins in the mouth with the aid of which enzymes? Well, ATP isn't an enzyme. Pepsin does not act in the mouth. Glucase, that would be not an enzyme that acts in the mouth. Neither is NAD+, that's a coenzyme. So the answer is actually amylase, and you probably heard me talk about salivary amylase. For example, starches begin to get broken down in the mouth by salivary amylase. So the answer is amylase. Number 20. What term describes the synthesis of glucose from non-carbohydrate starting materials? It's definitely not glucosidase. The next one is gluconeogenesis, right? If you think about this word gluco, meaning glucose, right? You have kind of glucose, and then you have neo, which is new, and genesis, which is, making. So yes, the answer is gluconeogenesis. If you look at glycolysis, that's the anaerobic catabolism of glucose. The next one is glycogenesis. That's the synthesis of glycogen. And glycogenolysis is the hydrolysis or the degradation of glycogen. So the answer is gluconeogenesis, a way to make new glucose from non-carbohydrates. Number 21, what hormone is released from the pancreas in response to low blood sugar levels? Well, it's not insulin, because insulin is released in response to high blood glucose levels. If you have a friend who's diabetic, you know, they go to a fast food restaurant, they probably give themselves an injection of insulin. So it's definitely not insulin, but the hormone that's released in the pancreas in response to low blood sugar would be glucagon, because glucagon is responsible for breaking down, we'll put here, breaks down glycogen, right? And glycogen is a polymer of glucose, and that's the primary storage of glucose in the body is in glycogen. And so glucagon is that hormone that responds to low blood sugar or low blood glucose levels. Same thing. Number 22, what polymer is the principal storage form of glucose? Well, I just went over this with you. The answer is glycogen. Glycogen is quite similar in structure to amylopectin that we looked at in the class. Amylopectin has alpha D glucose linked together with alpha 1,4 linkages, as does glycogen. They both have 1,6 branching. The difference is that the branches in glycogen are shorter and they are more frequent. So shorter and more frequent branching in glycogen. Number 23, which of the following statements concerning the digestion of food groups is false? This should be fun. A, proteins are broken down into amino acids. Well, that's certainly true. Digestion of all food groups begins in the mouth. That is false. Okay, that is definitely not true. If you think about fats, the degradation of fats begins in the small intestine. Small intestine, right? They get acted upon by bile salts, and they get, converted into these smaller droplets, and so more surface area is exposed, and then they can be acted upon by pancreatic lipase. Okay, if you look at C, carbohydrates are broken down into glucose. That's certainly true. Polysaccharides are broken down into glucose. Absolutely true. Fats are broken down into glycerol and fatty acids. Absolutely true as well. 24, what is the pentose? Why is the pentose phosphate pathway important? I told you we call it the PPP, the pentose phosphate pathway. And I told you that our book has very little information about the pentose phosphate pathway, but I told you that you should know it's got something to do with NADPH, right? It's not only an alternate pathway for glucose oxidation, but it provides molecules like NADPH, which are required for metabolism, and it also provides some sugar phosphates. And I went over this with you in our last lecture. And so this is the answer right here. Just, in this class, if you look for NADPH, it's oftentimes going to have something to do with the pentose phosphate pathway. Number 25, what substrate is converted into G6P or glucose 6-phosphate by the enzyme hexokinase? This is the first step in glycolysis. So first step in glycolysis, and you should know the step that we take a glucose and then it combines with ATP. The enzyme is hexokinase, and we end up producing G6P, which is glucose 6-phosphate, G6P, and we end up producing ADP and a proton. Anyhow, the substrate that's converted into G6P is obviously glucose. That wasn't meant to be anything more complicated than that. The glucose is the substrate. Okay, number 26. What is the final product? Into what final product is glucose converted at the end of glycolysis? So our glucose, once glycolysis is complete, it's converted to pyruvate. And the structure of pyruvate looks like this. If we have a methyl group and then a carbonyl, and then we have a carboxylate like this. This is the conjugate base of this acid, which is called pyruvic acid. Anyhow, so pyruvate again, the conjugate base of pyruvic acid. When you hear 8 in chemistry, you think of negative charges: sulfate, nitrate, carbonate, phosphate, pyruvate. Okay. Anyhow, there we go. Number 27. Which of the statements concerning gluconeogenesis is false? Our book goes into a little bit of detail about gluconeogenesis, but one of the most important things that you got to know about gluconeogenesis that we went over in the last lecture is that gluconeogenesis is not simply the reverse of glycolysis. It's close, right? It's close, but we know that there are differences at steps 1, 3, and 10. Those are not the reversible steps. In fact, the last step, there's even sort of an extra step there in gluconeogenesis. And you don't have to have these all memorized. But what you should know is that gluconeogenesis, we're looking for something here that's false. Okay, if we look at the first statement, it says gluconeogenesis is a pathway that is utilized when vigorous activity depletes blood glucose and glycogen stores. Of course, we'll use gluconeogenesis, especially in like starvation situations or again vigorous exercise or activity. Let's go to C. The level of carbohydrate available from the diet controls gluconeogenesis. The gluconeogenesis pathway is active for a diet low in carbohydrates and inactive for a diet high in carbohydrates. That makes sense. Gluconeogenesis is the process by which glucose is synthesized from carbon atoms from non-carbohydrate sources. That, in fact, is a great definition of what gluconeogenesis is. Gluconeogenesis is a pathway that protects the brain and red blood cells from experiencing a loss of glucose. Absolutely true. The one that is false is B. The gluconeogenesis pathway is the reverse of glycolysis. It is not the exact reverse. And in fact, like I just told you, steps 1, 3, and 10 are not the reverse of the steps that we see in glycolysis. Number 28. Which of the following is a true statement concerning the breaking of the phosphoanhydride bond of adenosine triphosphate to form adenosine diphosphate? Well, the first one made me laugh when I looked at it. It says hundreds of kilojoules per mole are released in the form of heat. That's not true. Okay, that's definitely not true. When we break apart ATP to produce getting ahead of myself here, to produce ADP plus a phosphoryl group, okay, we release 7 kilocalories per mole of energy. So it's not hundreds of kilojoules per mole, it's only 7. Water is formed because the breaking is a hydrolysis process. No, we're not forming water because we're breaking a phosphoanhydride bond. It's an endothermic process. No, it's exothermic because we're releasing energy. Exothermic, okay, energy is released, which can drive biological processes. This is absolutely true. Okay, that hydrolysis of that ATP, we can harness this energy in order to drive biological processes. Let's look at 29, which uses the exact same equation, just written in the reverse, right? I had it written here. Okay, if I had written it with the equilibrium arrow, it would be the exact same thing that you're seeing right here. They just show the full structures, and it's in reverse. Which statement best describes how the interconversion of ATP and ADP is responsible for storing and providing energy for cellular reactions? A, energy stored in ADP is released when ADP adds a phosphate. That's not true. Energy is stored in ATP. Energy requiring reactions of the cell can drive phosphorylation of ADP. No, they're going to need energy, and phosphorylation of ADP also requires energy. C, energy is stored in ATP and released when ADP undergoes phosphorylation. Well, the first part is true, right? Energy is stored in ATP, but it's certainly not released when ADP undergoes phosphorylation. It's when ATP undergoes dephosphorylation, right? Then you release energy. Energy is stored in ADP and is released when the phosphoanhydride bond of ATP is broken, right? Broken. That's what I call dephosphorylation, and that is true. So when you break that phosphoanhydride bond in ATP, you release energy. The last one, energy is stored in ADP and released when the phosphoanhydride bond of ATP. That doesn't even make any sense. Definitely not. All right, what is the function of coenzyme A? And you probably noticed it in our book, her abbreviation for coenzyme A, which she has the whole structure of coenzyme A in there, and I have it included in my videos. But coenzyme A is a thioester, isn't it? You have your acetyl group, and then you have the sulfur atom. So this is a thioester, and then you have coenzyme A, which is a big molecule. But what is the function of that molecule? To oxidize carbohydrates in glycolysis? No. To carry ADP to the citric acid cycle? Definitely not. But to carry acetyl groups in the form of acetyl coenzyme A into the citric acid cycle. That is, in fact, the purpose of coenzyme A. And you should remember that this part here of the molecule, this is an acetyl. That's an acetyl group right there. And the rest of it here, this would be the coenzyme A. Anyhow, so that's the function of acetyl coenzyme A. Number 31, in which stage of metabolism is the most energy in the form of ATP produced? Well, it's definitely not in glycolysis, right? That's an ancient pathway which provides very little ATP. It does provide some, but very little compared to oxidative phosphorylation, right? This is where we get most of the ATP that's produced. It doesn't come from glycolysis or the citric acid cycle. In fact, most of it comes from oxidative phosphorylation when you have protons going from the F0, F1 complex, and then we end up phosphorylating in order to produce ATP. Number 32, what cyclic metabolic pathway is the final stage of the degradation of carbohydrates, fats, and amino acids released from dietary proteins? Well, all of those carbohydrates, fats, and amino acids, they can all go into what cycle? The citric acid cycle. In fact, if you look at our lecture video from our last class, we looked at an overview of catabolism that had this figure on it that showed amino acids, proteins, or sorry, amino acids, rather, carbohydrates and fats, and it said, well, all of them can end up with intermediates in the citric acid cycle. Number 33, if you look at the F0 F1 complex, what is the role of the F0 portion of that complex? And if you look at the structure of it in the figure that's in our textbook, I'm not much of an artist, but there's a really great picture of the F0 F1 complex. It kind of looks like this. You get this portion here, and then she has F0 written on that. And then the F1 complex kind of has two pieces like this. Then you get, 1, 2, 3 parts like that. And then it's kind of got like 1, 2, 3 things at the bottom. Again, I'm not a very good artist, but this portion down here, this is the F1 part right here. And the protons, so the protons end up coming through the F0 complex, and then they get into the F1 complex, which of course causes, I'm running out of space here, but it causes the phosphorylation of ADP. So we have the inorganic phosphate, and we produce ATP. And so the function of the F0 portion is that, where was the answer? Here we go, it's C. It provides a channel for the return of H plus or hydrogen ions into the matrix, because this complex, this is the wall here. Anyhow, I'm not much of an artist. Okay, so you have the inner membrane here, and then you have the phospholipids. Okay, like this. Okay, so this is the matrix in here. And this is the inner membrane, like that. And then you have the intermembrane space up here, intermembrane space up there. Anyhow, that's the whole point behind the F0 portion of the F0 F1 complex. All right, let's move on to number 34. Which statement concerning the regulation of the citric acid cycle is true? Well, if you read through all these, you take the time to read them, you think about the citric acid cycle, right? We're talking about making energy. First answer, high levels of ADP, which is a precursor to ATP, stimulate the reactions of the citric acid cycle. Well, that's true. If you have, an abundance of ADP, that's going to speed things up. High levels of NADH inhibit the reactions of the cycle. Well, yeah, NADH is a product of the citric acid cycle, so if you have high levels of it, it's going to inhibit it. Low levels of ATP stimulate the reactions of the cycle. Of course, if you get low energy, yeah, it's going to get the citric acid cycle going. And then high levels of ADP inhibit the reactions. That also makes sense because it's going to be like a negative feedback inhibitor, right? If you've got enough ATP. And so all of these statements, in fact, are true. Number 35 is kind of a memorization one. You'd have to familiarize yourself with the transamination reactions. I have a whole short video about transamination reactions. I forget exactly which section it is of chapter 22. 22, oh, I can't remember. Degradation of amino acids. So this is section 22.7. Okay, I have a whole video about this. And the coenzyme that's involved in all transamination reactions is pyridoxal phosphate. And I go over how pyridoxal phosphate works in transamination reactions in that video. Number 36, the oxidative reactions of metabolism provide for most ATP production. In what specific part of the cell do these reactions occur? Well, if you're talking about ATP production, you are talking about the mitochondria, right? Sometimes we call this the powerhouse of the cell. This is where all the energy is produced, is in the mitochondria. And in fact, if you look at chapter 22, video 22.1 just deals with the mitochondria. In fact, it's the first part of the aerobic respiration chapter. Number 37, what is the usual fate of the alpha amino group in the catabolism of amino acids? So going back to the degradation of amino acids, if you go to section 22.8, it deals with the urea cycle. So those amines, or those amino groups in amino acids are converted to urea. So it is converted to urea, which is excreted by the body in urine. Number 38. What is the term for the region of a mitochondrion that is enclosed by the inner membrane? And I told you that that is called the matrix space. So the matrix space. If you go back to my video on the mitochondria, I explain a little bit about the structure of the mitochondria, and then I go over how the portion that's enclosed by the inner membrane is the matrix space, and the part outside of the inner membrane is the it doesn't really have a special name, I guess. Sorry, what I was thinking of is the outer membrane. So we have the outer membrane, then we have the inner membrane, which is all folded. You have the cristae, and then the inside of that would be the matrix space. Anyhow, I'm making it more complicated than it needs to be. At least that's what I'm doing in my mind. Anyhow, let's move on to the second to last question. Where does the degradation of amino acids occur? This, again, is covered in the section on the degradation of amino acids, which is video 22.7. And the degradation of amino acids occurs in the liver. The last one is, what is the terminal electron acceptor in aerobic organisms? The answer is oxygen. If you look at the oxidative phosphorylation, if you look at figure 22.7, so not section 22.7, but figure 22.7, it shows you what happens to the electrons. And the balanced equation is if we take half a mole of oxygen plus 2 protons plus 2 electrons, we end up producing water. And so oxygen is that electron acceptor. That covers the entire Practice exam 5.